from Microbe TV. This is TWiEVO, This Week in Evolution, episode number 116, recorded on July 23rd, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from LD Lab Studios, Nels LD.
Hey, Vincent, good to be with you. And yeah, we changed our moniker up there. It said the University of Utah before, but I've been, we in academics, as you well know, Vincent, we undergo our annual trainings where we're, brought up to speed on exciting topics like biosafety and HIPAA sort of considerations, things like this, kind of dotting the I's and crossing the T's. One of the new ones they've added to my menu is the idea that I don't represent the university in an official capacity in some freedom of speech related issues that somehow got garbled in there in ways that still are. I guess I'll have to engage in some remedial training in years ahead. But I'm I'm very happy to be with you from as always from LD Lab Studios to share my ideas and insights, whether or not the university wants to endorse them. I would take a moment, though, to endorse the University of Utah. I love working here, the colleagues around me, the sort of work-life balance, sort of living the scientific life in the Western Rocky Mountains is pretty spectacular. So I'm a fan, even if that's maybe not always sort of reciprocated by my official institution.
I never really associate myself with the university. People know where I am, but I'm at Microbe TV.
That's right. You're now a free agent in a sense. You've.
I mean, I still am a professor at my university, but I never featured them prominently. Even when I started TWiEVO back in 2008, it was just TWiEVO, and they never complained. So,
yeah,
I don't represent them. I represent myself, and Nels represents his self too, himself.
Exactly right. Yep.
Welcome, everybody.
Yeah, we pulled an apologies. So we pulled a little bait and switch. This is my fault on our timing here. We're coming in a couple hours late, so I'm engaging. Actually, in this case, not remedial training, but mentorship training, which is I don't know, the summer season. This is usually when traditionally professors are meant to be lazy or take it easy. For me, somehow the reverse is true. It feels like the temperature, the volume gets turned up on sort of commitments and so forth. We're also, Vincent, I think this is going to be an express edition of TWiEVO. We're going to sign off a little before 4:30 Eastern. Is that? Yep. And so I think, but we'll fit in a lot of good conversation and good science in the time that we are sharing together.
Yeah, I got Daniel Griffin coming up in an hour, and then I gotta head home so I can do tonight's live stream.
That's fantastic. So, and did I see that Angie Rasmussen is going to join you on the office hours?
Angie's going to be my guest tonight, my former student and a spokesperson for science who many people have heard. She's very vocal, so should be fun.
Fantastic. I think that will be interesting. Tune in.
So we have a couple of moderators here today. We have Barb Mack UK, we have Les, and we have Vanity Nutrition. Vanity, are you in Montana already? You're making some dental floss? Let us know. Vanity was heading to Montana for medical school.
Oh, very cool. Yeah.
Switch jobs.
Thank you all for being here.
Thanks for being here, everyone. Let us know where you're from. We can shout you out here. Let's see. Barb Mack, of course, is in the UK, where it is currently dry, cloudy, and 17C. Jeremy's in Seattle, 76F.
Welcome, Jeremy.
Red Desk is in London, who has his own MAGA wannabe trying to emulate your orange god with xenophobia, anti-vax, anti-women in the police, etc. Yep, that's not good.
Long, hot summer indeed.
Abdullah X, where did Racaniello? Little botch on the spelling there, man. But it's okay. Where did you get your statue of poliovirus with, where is it? Here, right up here, with the receptors slotted in the grooves. I had made a company called Molecular Designs, I think, made it for me.
Very cool.
It's not cheap, folks, but I can give you the contact information. Just send me an email, Vincent at microbe.tv. I bought it.
Welcome, welcome, Abdullah.
No, and it's okay. Don't worry about it. No worries. I don't mind when people misspell my name. It happens. Gary's in Denver. We're at 79F in that corner of the TWiEVO verse.
I love it.
Jill is in Kansas. Welcome, Jill. Anybody else want to fess where they're from?
Tight knit group. Appreciate you all being here. And
Yeah, we appreciate it. We have an interesting paper today. Which,
yeah, why don't we jump right in?
Right in. Go ahead.
That we're
By the way, if you like these programs, we'd love your support. We don't do ads. We depend on you, and we give you in turn good science, great science, and all truth, no lies. Microbe.tv/contribute. Nels, we like ants here on TWiEVO, don't we?
Guilty as charged. I think there's like there's some clues in here about maybe a former scientific life. Maybe I was an ant researcher or something. I can't resist these papers that involve ants as it relates to sort of especially some of the charismatic behavioral traits that we see in some of these insect societies, these eusocial critters, and then the sort of prospects both for doing experiments to try to get at some of the genetic basis of behavior, but then to really put it into that evolutionary context to think about how did these things originate? How did this sort of complexity arise? And are there sort of clues using the tools that are sort of coming online, even at genomic scale, to think about some of these things or to understand them in new ways for the first time? So, as Vincent said, we're aiming for the truth. I don't know if all truth and no lies, all the science truth and nothing but the truth. We'll do our best, of course. Approaching the truth from a scientific perspective always comes with its caveats, etc. Okay, but anyway, so let's jump right in to this paper. The title of it came out about a month ago. The title of it is "Neuropeptides Specify and Reprogram Division of Labor in the Leafcutter Ant, Atta cephalotes." And a cast of characters here from the University of Pennsylvania. Shelley Berger's lab is Shelley Berger, the senior author. 3 first authors. This is Michael Gilbert, Carl Glastad, and Maxim Fioriti. Along with, I think, some collaborators, for example, in Switzerland, in Seattle, and St. Louis. Or again, all points on the map, the way modern science is often practiced. And then another feature that has sort of come on our radars, or we've added it to our radar, given some of the headwinds as it relates to practicing science today in the U.S., is to think about where did the funding come from for this work. And so a lot of NIH grant funding and evidence here. So both the first authors who had or have fellowships from the NIH, these are training fellowships, the so-called F32 mechanism, the F31 mechanism, both as grad students and postdocs. There's R01 grants from the NIA, which actually, you know offhand which national institute that is, Vincent? Is that
what's the address?
NIA, R01.
NIA.
I don't know about that one. Maybe this is a typo. There's also AG and NIMH, which is mental health. There's an R01 there. And so, 3 R01s, 2 fellowships. And then in order to get some of these ants, these leaf cutting ants that we'll be talking about in detail, imported from Trinidad and Tobago, where they actually are cutting leaves and behaving, this involved permits with the USDA. And so there's a whole scientific infrastructure kind of percolating here under the surface that in many cases is really in jeopardy as we think about science going forward. And so trying to just add that to, usually it's kind of a footnote, but now it feels sort of especially critical to keep that sort of on the in our sights here.
Now as I'm looking up on the NIH website, I don't see what AG is.
So there's of course anything with the government involves a lot of acronyms, and so mental health, that one seems familiar.
GM,
NIA,
General Medical
GM is
Sciences.
one.
Yeah.
Yep. NIAID is one that you and I are pretty
No,
familiar with.
it's aging.
Aging. Yeah.
Yeah, AG is aging.
Mhm. And so, yeah, all sort of the connection between scientific research and how we fund it, sort of a story underneath the story here. Okay, but so,
Nels, look
yeah,
at this. Isn't this graphical abstract great?
it's fantastic. Yeah, so we've got these cartoon ants. This actually, we can almost tell the whole story with the abstract here in some way. Maybe we'll kind of give away the punchline. So this is these castes or these society, these sort of division of labor that you see in a lot of eusocial insects, Atacephalotes, the leaf cutting ants included, or the species of them. And so they've, and they'll do some work here that will describe categorizing the ants sort of by their morphometrics. And so we're seeing this here, the ants that have the large heads and sort of this embellished strongman tattoo. This is a major of the major caste involved in defense of the colony. You've got these media or medium-sized ants that are involved in leaf harvesting. Smaller ants are the minor caste involved in caretaking, and even smaller ones, these farmers that are involved in caring for the fungus. So it turns out, I didn't know that took some time for me to learn this. I think a lot of us assume that the ants, when we watch them on the nature shows as they're collecting, cutting the leaves and bringing them to the colony, that they're eating the leaves. That's not the case. They're actually cultivating fungal microbes, fungi, on those leaves. So the fungi eat the leaves and the ants eat the fungi. It gets complicated in a hurry, including.
So this is interesting. This big one, the major, is involved in defense. But look, it's got a mom tattoo on it. I have to blow it up here, right?
That's
and
right.
then the thing is, as you will tell us, you can transform this big hulking defense into a caretaker.
That's right.
Or a leaf harvester. But you know what's weird? The caretaker lost the tattoo when it should have kept it, and now the leaf harvester has the mom tattoo. But that's cute, isn't it? And then now the caretaker has a little baby carriage. That's so cute.
Pretty good. How the tattoo got transferred, that's maybe I don't know if we'll solve that one on today's episode, but yeah, you're right, Vincent. So we've been talking about ants here on TWiEVO for years, stretching back near the beginning. I still remember this really fun conversation we had with Corey Moreau, an expert in ant societies, ant biology. And in fact, we even called the episode "This Week in Ants." I think there was a moment where you were considering launching an entire podcast on the topic of ants, given the fascinating biology under the hood here at sort of all levels of sort of scientific inquiry. We weren't done, so we interviewed another leader in the field of ant biology. This is Daniel Kronauer. He had just published a book, Army Ants, which is beautiful. So he took many of these photos himself and then tells the story again of some of these complex behaviors, these societies, through the lens of someone who really cares for these things and just has dedicated his career, his life to better understanding some of these, including pioneering some of the molecular techniques to be able to set up experimental manipulations. And so returning back to TWiEVO episode 18, Raiders of the Lost Orco, still one of my favorite titles ever that you came up with. These were some labbies, grad students from the Kronauer lab who were telling us about how they were doing genetic manipulations of raider ants in this gene in particular, Orco, to sort of glimpse at the possibility of sort of using genetic manipulation to impact some of the behaviors or outcomes with the ants that will sort of pick up the chase here a little bit with today's work. Also recently, I actually forgot about this only a few months ago, on TWiEVO 110, we picked up a paper, the title of the episode, Ants Go from Carpenters to Surgeons, with evidence that these ants were actually performing amputations on their colony mates, depending on the type of injury that they had. And so another sort of illustration of like all of this complex behavior that's just like right before our eyes or under our noses when it comes to the ants. And so here we're back at it with a different set of species. So this is Atacephalotes, the leafcutters, which are
back at it. Very good.
There you go. Didn't see that one coming. Um, and again, sort of taking some of these behaviors, but then asking if there are sort of genetic manipulations that are possible to get at a better understanding and even the origins of this. Surprise! It's not in that graphical abstract that you flashed up there, Vincent, which is, I agree, is pretty great. But there's going to be a surprise appearance from naked mole rats actually later in the paper, which kind of comes out of left field to say the least. But we'll kind of weigh that as an interesting part of this work as well. Okay, so a little bit of background on the leaf cutters, and I think we're probably have all seen them on those nature. They're so charismatic on those nature shows where they're kind of lined up, and you see that parade of ants that are bringing these, perfectly cut leaf pieces in their jaws and bringing them into the colony. And then it gets even more interesting as you go sort of below the ground and see that they set up these gardens where they're cultivating fungi, and then all of these layers of nursing or taking care of their food sources. So treating the fungi with antibiotics. There's just so there's like bacteria involved. There's fungi involved. There's communication using small molecules between the ants as they sort of execute on these stable societies, and then they're taking care of each other, right? So as you pointed out, they're defending the colony. Some are in these divisions of labor, so all the genomes are the same, right? But you've got the queen kind of at the center of the action, reproducing, and then these very different forms that are out performing these different tasks. And so an sort of long-standing classic question is, how do you evolve that when you have the same genome, or how do you regulate that? And that's sort of where we pick up some of the action in today's paper. So same genomes, different behaviors. How is this regulated in particular at the genomic level? And so, the paper, I have to say, I'd love to get your opinion too, Vincent. It's pretty dense. They cover a lot of ground here, and even we'll bring in the naked mole rats. And so we'll probably not do justice to all of the details going in here, including, at least for me, that jumping off point. So when we're writing or sharing our work after, obviously in this case, years of efforts, how to tell the story, or how did you get on? So the main focus of this work is two specific neuropeptides that they're going to make the case or provide some evidence that are really involved in some of these behavioral outcomes, these division of labor. And so they kind of just drop us in the middle. We kind of parachute into the middle of the story almost, where it's like, okay, well, here are the two that we've decided to manipulate. And I should maybe back up a little bit. They do some, initial characterization, or describe some initial characterization, where they're trying to classify in ways that are useful to link or correlate this to some of the genomic patterns between the groups. Well, what are these groups in the first place? And so they use, or they propose what we already kind of glimpsed at, these four morphometric categories: the major, the media, the minor, and the minimum, the little garden tenders, fungal garden tenders.
By the way, Nels, which one would you want to be?
Easy. The garden tender, I love gardening actually. And so this notion of cultivating fungi, I mean, I kind of do that as a researcher too. And when we study, baker's yeast, Saccharomyces. But what about you, Vincent? Would you be the big tattooed defender, or do you fall into the cast?
I'm not a violent person. I don't like conflict. I'd like to be the leaf harvester.
Yeah, that's pretty good.
Because you get to go outside instead of staying underground all the time in that ridiculous subterranean home. You go outside, you get some fresh air. Of course, there's a risk that a bird's going to eat you, but that's nature.
Yeah. No, fair enough. I think that matches. You're also that you're in front of the cameras, right? When you watch those nature shows, it's the leaf cutter is up, like you're doing the work in the front of the cameras, and I think that fits very nicely with the scientific efforts that you practice as well. I like to hide out in the lab, kind of make myself as invisible as possible, and just kind of think about how do we grow stuff that is tasty and nourishing, I guess. But anyway,
By the way, I just wanted to point out that Luis has got it. The ant epigenome. That's what this is all about, right?
There you go. That's right. And that's right, Luis. So how do you, with the same genomes, how do you sort of write these very different biological stories that sort of reflect these different behaviors, and even echoes to some of our own, as scientists, our own sort of styles. We can almost, I think that's what's also really sort of compelling about the ants, is somehow you can kind of see yourself in some of the simple activities somehow. Okay, so they use morphometrics so the head size of the heads to sort of define these, and then to correlate that with those behaviors that we've been sort of hinting at. And then they start to put this together. This is where we kind of parachute in the middle of it to focus on two neuropeptides that they think are impacting that. And by the way, neuropeptide biology, absolutely fascinating. These are small proteins, amino acids linked together. And there's not, in a lot of cases, the neuropeptides are short, but they pack a punch. They pack a biological punch. And so a long history of sort of central roles impacting or influencing behavior as it relates to insects. And so they aren't starting the project out of whole cloth, and they're candidate hormones, or neuropeptides, hormones in this case, that they sort of have that clue that these might be sort of the right places to focus.
By the way, I just want to point out that they have what they call an arena where they duplicate these different activities, right?
Yeah,
and
super important.
They could watch the ants do defense or farming or whatever, and then they can take them and do RNA sequencing and see what genes are induced, right? And that's how they get to these neuropeptides that Nels is mentioning. The neuropeptides pop up as a candidate for, there's some consistent neuropeptides that are specific for each behavior or caste. And that's where we jump right into this story, right?
Yeah, absolutely. And I'm glad you brought that up too, Vincent, because, so the ants themselves are, in this case, the colonies that they're working with are native to Trinidad and Tobago. But then when you and exhibit all of this complex behavior, even more complex, right, in the wild. And so then as researchers, as experimentalists, one of the challenges is to sort of try to recreate that, but in a laboratory setting where you can, have the ability to measure or manipulate in ways that are really challenging in the wild. And so,
And so this arena is a cool conception. They do videos, and then they use assays to identify the majors in the media and so forth, and they take those and extract RNA from them.
Extract RNA, and then you end up, with transcriptomes, right? So that's the sort of molecular correlate that they're making their comparisons to. Really challenging. So, because you've got, so in this case in particular, they're looking at differential regulation of genes, right? So the genome is the same, as Luis pointed out, but the things that are expressed, depending on, what member of the caste that is, if it's a defender or a gardener, et cetera, or a leaf cutter, the different genes are turned on. And so still you're sifting through thousands of them. And then I think putting that together with the candidate genes with sort of already what's known to nominate these two neuropeptides as sort of candidates that might be sort of really influencing some of the biology here. So the two neuropeptides in particular, this is fascinating. So there's first one is CCAP, that's short for crustacean cardioactive peptide. And this one will turn out to its presence or absence impacts whether the ants are likely to exhibit behaviors that are consistent with foraging for leaves. So the kind of leaf cutting, the media caste members are, showing a lot of this behavior. And again, this is in its mature form. This is a 9 amino acid peptide with a 1 disulfide bond, and a very small amount of sort of biological information that has a big impact on all the way to the behavioral level. The other neuropeptide is called NPA for short. We're in the alphabet soup here. This is neuroparsin A. I think both CCAP and NPA are insect specific. That becomes an important detail, especially as we'll circle back to some mammals, the naked mole rats. And this one larger, so 83 amino acids, dozens of cysteines, and somehow in that, so cysteines are involved in these disulfide bonds, these post-translational connections between the way the peptide will fold up into a 3D structure, and then that influences its function as well. And NPA has been proposed to impact, or they'll show some evidence that this also impacts brood care. some of the behaviors that you see exhibited in the minor caste of the colony. So, okay, so we've got the four castes, their morphometric definitions, and already the lab arena, as you pointed out, in order to capture some of this complex biology, to record it, to characterize it, and then to correlate it, the behaviors with the molecular details or the transcriptome. So the transcriptome, this is, we're pretty comfortable with genome as being the collection of our chromosomes. The transcriptome is the collection of our transcripts, or the RNA that gets transcribed from our DNA. And then to use the tools of genomics to just capture all of those from a sample. In this case, the samples are the heads of the ants, or collecting the brains. And then asking, as you collect these things, as the ants are behaving. And actually, this is an important, not just a detail, but sort of an important part of the experimental design, is they're actually using in their arenas, they're trying to capture the ants sort of in the process of exhibiting the behavior that they're interested in order to get transcriptomes, to try to get a sort of an echo of real time of what's sort of the biological basis of what's going on here. Now, I think we can, argue or quibble about, is this phenotype or this outcome of measuring transcripts really capturing all of the biology here at the molecular level? Some of those transcripts might become proteins, others may not. There's sort of all this other, these details of how the biology is going on. But this is something you can measure and compare reproducibly. And it's also, there's a lot of information in there, how to gain understanding from that. I think that's sometimes the bigger question at hand. So just thought I would put that out there. The early returns on the analysis is that for each of the classes, so I think this is now divided into those four classes, they see about 1,000 uniquely upregulated genes. And they put that into these categories. And so, they take those thousands of genes and then try to categorize them for their functions, which is sort of a standard level of analysis that goes on. And then what they come up with is that peptide hormone secretion is one of the categories that they see a lot of differences between the different ant groups, and that sort of ties together with some of the past work in the field as well. I'd say, to the author's credit, they do a little mass spec. So they take ant brains and they ask, not only do you see those transcripts, but are those proteins that are, so those transcripts then get translated into proteins, are they there sort of in quantities that are consistent with the transcriptomics? And at least for the sort of peek under the hood they did with mass spec to measure protein presence and kind of quantities, I think they would argue that at least it's consistent with what they're putting forward here. And then, so two stuck out. So in the kind of categories of differentiated genes, as it relates to these neuropeptides, CCAP was in the media ants and NPA was in the major ants. I think I said that was in the minors, but it's actually, they first spotted it in the major ants. And here we get the first hint of kind of a comparative approach. So in addition to thinking about the very specific species here, they kind of looked at data from, or generated some data from another ant species. And I don't know all the details here, or some other insects. It might have been some data that was already published, and just sort of kind of some gut checks about, do they see some of the same patterns emerging in the differences in the transcriptomes from some of the other species, which I think is the right thing to do, sort of how to interpret that. So how much consistency do you expect, or what's different because the species have diverged or sort of rewired some of this biology, that harder to kind of discern the details of that or the value of that sort of in this setup. But again, the paper covers a lot of ground, both to their credit and for the reader, it makes it a little hard to sort of parse through what the important parts are. But all of this, Vincent, I would say, sets the table for something that they can do in leaf cutting ants, which you can't do in a lot of wild species out there, which is some genetics to manipulate the system in a really kind of taking these genetic scalpels to it and then asking, if we mess with things, what happens? This is sort of maybe part of, our fascination with ants. Like, goes back to when we were kids, messing around with magnifying glasses and burning them. This is sort of like a sophisticated sort of, way of sort of recapturing some of that curiosity or something. So here, what they're doing is not burning them with magnifying glasses, but they're knocking down either of those neuropeptides, the CCAP or the NPA, in specific cast members, right? And then asking how does that impact the transcriptomes that they recover from them if they've either knocked down, taken away to a large degree the CCAP or the NPA neuropeptides, or overexpress them, inject those peptides into the ants. And I have to confess that I haven't gone deep enough in the paper. Did they inject the peptides themselves, the proteins?
Yeah.
Or they did, yeah.
So they knock down the RNA. You can just inject double-stranded RNA, and it will knock down the target. And then they inject the peptide into the brain of the ant. I don't know how they hold them. Maybe they anesthetize them or something, but it's really cool. And they have ideas about, so the peptides can do a couple of different things. They could induce a behavior or repress it, right? And so they have hypotheses about how that works. And for NPA, they also have its putative receptor, which is called VKR, and they can knock down that too, which gives you a double confirmation. It's very cool.
Yep. I agree. And that's a really nice part of the work. It shows, I think, the extent of the work, that they don't just do the peptide, they do the receptor as well, and sort of have that kind of call and response almost. Yeah.
The other thing that's very cool is that the ants don't change morphologically. They just change their behavior.
Correct.
Right? So this is that's why this big hulking defender ant, you can change it into a caretaker, but it's still big and hulking. It's so cool.
Right. It's very cool. I think that raises another sort of critical point here, right? Is anytime you're manipulating like this, you're always kind of thinking in the back of your mind, okay, well, how specific was that sort of, intervention? So worst case scenario, right? If you're really bad at injecting peptides, you might just injure the ant so badly. And then, if you're sort of interpreting, well, the ants all got kind of sick and lazy, maybe that's just because you were really bad at injecting them. But here, right, they've got the sort of behavioral correlates so well defined that I think there really is signal there. Now, whether or not they've, whatever transferred tattoos and the entire sort of set of behaviors versus, again, sort of the subset of phenotypes they measure. So here the data is read out, I think, in ways that make total sense. So how often do they move? So after the experimental treatment manipulation, how often do they move brood around, right? So they're watching them experimentally, they're counting that and then correlating that to the overall behavior. But I think I would say that they've pretty convincing evidence that these numbers are moving and the behaviors are different. How that relates to the sort of suite of the entire behaviors is still sort of an open question in some cases. But the fact that, again, that they kind of go through the effort of not just doing the neuropeptides, but also the receptors, et cetera, and start to be able to kind of see whether those results are consistent, really does bolster.
So why don't we summarize what happens with all the peptides?
Okay. You kick us off, Vincent.
So you start, if you knock down either NPA or its receptor, VKR.
Yep.
The majors, the major leagues here, the majors, they move to caretaking.
That's right.
Okay. So they think that this is a key peptide involved in caretaking. Again, the morphology of the ant doesn't change, but it becomes a caretaker. And the receptor, knocking down the receptor has the same effect, which further supports VKR as a receptor for NPA. Now, the other idea is that NPA suppresses caretakers in media, right? And media and majors have high NPA. And so they also did that in media, and it knockdown of NPA reduces brood movement by media. And so the NPA governs caretaking across the castes. When they inject the recombinant NPA into the minor brain, this reduces brood movement. Yep. So that's why they have high NPA to suppress the brood movement part of that behavior. And that's conserved across all the ants. The other thing that these majors do a lot is bite. That's that's one of their defensive moves. So they ask whether NPA can promote biting. And in fact, when they reduce MPA, it promotes caretaking and reduces biting. So NPA is suppressing biting. It's very
That's
cool.
right. I agree. Very. Yep.
Then the
Yeah.
other one, of course, is CCAP.
Hmm.
They inject this into major and minor brains. These two don't have leaf move. They don't have leaf movement. They don't carry leaves. All right.
Hmm. Hmm.
When you inject the peptide into majors and minors, it makes them get leaves. They
They start
acquire
moving.
leaf
Right.
movement.
They start
That's so
moving leaves
cool.
around.
I
Yeah, I
mean,
agree.
this is a
Yeah.
behavioral change by a little peptide, What is it,
Yep.
20 amino acids or something like that?
Nine amino acids in a
Nine
disulfide.
amino acids. Holy cow. So basically, CCAP is a regulator of leaf harvesting behavior, right? So they can make all of these different assays of behavioral changes induced by giving the peptide or by removing it, right? And
Yeah.
so this begs the question, of course, how does this work? How does a peptide do this? And actually, we're not going to get the answer in this paper, unfortunately, because that's really the interesting part, but it's really complicated, right?
Yeah.
So
Great.
the idea, of course, is that when the peptide is present, it induces other genes that make a leaf cutter out of a major, right?
Yeah.
So,
Yeah.
and you can imagine that it probably involves a lot of other genes, and they don't know which ones they are, but they do transcriptomics, and they can show that the peptides have a specific effect on the transcriptome depending on whether you're a major or one of the other casts, which is presumably, they call it a major transcriptome, and
Yep.
the peptide can cause a shift to the minor transcriptome, right? Which
Exactly.
is
Yeah.
presumably buried in there are the genes that are affecting this behavior, right? Um,
Agreed.
but
Yeah.
but that's the question. What are these induced peptide induced genes doing to change behavior? And I don't know.
Yeah. I don't either. They lead us right up, and that's again sort of that challenge between what you can measure and then what you can understand. And so I think they convincingly showed us what you just described. And then this is where we take this sort of like massive left turn or surprise, like right when you might. So, and by the way, I'm not trying to like the work up till now is already, I think, a pretty major advance for the reasons
Yeah.
you were saying so beautifully. But instead of, and fair enough, as the hard work ahead is to really link that to an understanding of what is specifically being changed that then impacts those behaviors or influences those behaviors as it relates to the changes in neuropeptide levels. But here's where the naked mole rats show up. And so at first, I have to confess, I was a little bit like, wait, where did this come from? What's going on here? So naked mole rats also, a eusocial mammal, different behaviors, but obviously, they propose maybe some convergent evolution of some of these. So it's not the forecast, but there are things like foraging and brood care. There's also tunnel digging going on by these guys. And so they ask a pretty daring question, which is, are there sort of, as they look at their transcriptional profiles correlated to the ant behaviors, are there sort of echoes of that even in this totally different species that independently evolved? So is there some kind of molecular logic underneath this that might sort of echo or mirror this, or a bit, and this is honestly, sort of where the evolutionary, to hang this at TWiEVO instead of This Week in Ants or something like that, is that this comparison, I think, is what's pretty sort of creative and novel about this work, to ask, is there sort of, a way to advance our understanding by thinking about sort of what's shared even over these long time periods as it relates to the genetic basis of behaviors. So to get at this, they start harvesting the hypothalamuses of naked mole rats and analyze their transcripts, as to see if there are some patterns that match the transcriptomes in both the casts and then the ones where they've either knocked down or injected the neuropeptides into those ant heads. And so here, maybe we pause for a little bit of podcast peer review. The challenge of drawing this sort of apples to oranges comparison, in a sense, I think is really, an interesting challenge here. So are you really kind of matching behavior? Like, you could imagine just like all kinds of variables that are going to influence, sort of the molecular outcomes, like the age of the animal, the sort of timing of where or how they're behaving, how that relates to that, and how you sort of sampled either a hypothalamus or an entire ant brain. Is that, like, are these really fair comparisons? And for me, I don't know, actually, to be honest with you, except to say that they point out that they think they do see some sort of shared patterns
Yeah.
among the transcripts that they can sort of match to some of these behaviors. So what they note is a remarkable overlap despite 600 million years of
Yeah.
divergence, including ones that sort of correlate to when they knock down those neuropeptides, change the behaviors, and then see some of those same patterns of differences in the transcriptomes. And so we're kind of glossing over it. There's principal components analysis involved in this to try to sort of characterize the massive kind of genome scale data, the thousands of differently expressed genes, and then to try to kind of pull from that some signal to say that, hey, there might be some shared sort of patterns that are emerging from these comparisons.
What's
So I
interesting
don't
is that these peptides are not present in the naked mole rat, right?
No, these are insect specific or crustaceous. Even in the name, the crustacean cardioprotective or cardioactive, sorry, peptide. Yeah, and so the
It,
sort of the molecular players are necessarily going to be different,
yeah,
but
and
then
yet the transcripts seem to overlap, as you say. And in fact, they do an experiment where they add this NPA peptide to cells from ants or the mole rats in culture, and they get similar transcriptomes induced. So despite there not being a peptide, NPA, or a receptor, something is going on, right?
yeah, agree,
I don't
and
know
yeah,
how
that's
that
even a
works.
yeah, no, agree. That's even another sort of layer of the analysis here is then they go to the cell lines. And there you can say, okay, well, now you've really taken sort of the behaviors out of context because it's now not the animal behaving, but it's sort of the response at the cellular level in a homogeneous plate of a dish of cells. I have to say, to me, this was the most kind of creative part of the work, the most daring part of the work, and also maybe, I don't know, a little bit of a head scratcher of, am I really buying it? I'm curious what you think, Vincent, about those comparisons they're drawing here with the naked mole rat. Do you guess how much weight do you give to their conclusions that there really is sort of a signal here in the shared patterns of the differences of the transcriptomes?
I don't know how NPA can do something without
Yeah.
without there being a receptor, right?
Hmm.
Unless something else is acting as a receptor, right? So I mean, these transcriptome analyses are tough, right? But I think their conclusions are justified, and other people have looked at this data and in more detail than we have, and it's been published in a good journal. So I accept it. But of course, there are other experiments you can do to address what's going on in the mole rat if you wanted to do it. This is an ant lab, I guess, right? So they're not going to be doing that, but maybe someone else can take it up.
Yeah, well, and I think it took that left turn. I mean, as you were sort of driving into the next step, which would be, okay, you've sort of pinpointed the impact of these neuropeptides, and then how does that work, right? Like that, I think is definitely a direction that will be exciting to see where that goes in the
Yeah.
future as well. One of the good things about the discussion section in this journal, in Cell, in the journal in particular, has added this limitations of the study section, and they point out a couple. I think there's probably some more limitations that could be weighed here as we think about the difference between
Yeah.
sort of hints of patterns over transcriptomes and the complexity of neurohormonal signaling and neurocircuitry. I mean, this is all written as well on not just sort of the genes that are upregulated, but the neurons that are firing and the synapses, the
Sure.
how the signals are sort of received and interpreted. And so there are sort of layers of biology here that are also at play when we think about the behavioral outcomes. And so it's really fascinating. And yeah, no, again, to see sort of, I think the system with the ants just kind of delivering on these age old questions and sort of getting closer to the kind of experiments that could really kind of drive to that question that you posed. How does this all work?
You have some interesting comments on this.
Yeah,
Let's
let's dig
see
in.
here. So basically, Krister and Luis are talking about methylation being involved, right?
Hmm.
So here, if Krista is suggesting a pheromone is involved in methylation, that turns on the peptides in themselves. But what I want to know is how the peptides, the genes turned on by the peptides, right? Presumably they're, I don't know, Nels, what is the peptide doing? It hits a receptor, and then that must end up turning on genes, right?
Yeah,
Downstream.
correct. Through signal transduction pathways. So the simplest idea, there could be more going on here, but the simplest idea is that the neuropeptide has a receptor. The receptor then signals that, or as the peptide binds the receptor, that then changes the activity of the receptor so that it makes a signal in that cell. That signal is then transduced through the cytoplasm
Mm-hmm.
into the nucleus. Oftentimes, this is read out through the binding of other proteins to transcription factors that then turn on or regulate the expression of yet other genes, and transcription factors can regulate other transcription factors. And so that's where, yeah, we
Right.
kind of, I think the authors and us sort of glossed over. Well, the authors go into much more detail. I've glossed over a lot of those details. And in a lot of cases, they're just measuring that sort of the output, but there's all of this biology in between,
Yeah.
um, where a whole lot less is known, with a lot of opportunities to sort of understand this. But yeah, no, absolutely. There's an entire fields obviously dedicated outside of ants and mammals to how neuropeptides are acting in the brain, how this is regulating gene expression and some of those outcomes. And then so this is sort of then put into motion as it relates to these behavioral outputs for the ant colonies, which is
So
sort of connecting it in a new way.
Luis says mind controlled peptides, the power of behavior in primates. Yeah, but these are not primates. These are ants. So very old, ancient behavioral effects, which may have come down to us. Rob says which peptides control our behavior. There are many. There are many, but we can't do the same kinds of experiments with us. So we do them in models. And the ant is a great model because you have these casts, right, with the same genome, and that's the whole point of this model. It's beautiful.
Yeah, yeah, yeah.
Okay, Nels, we're at 417. Should we do some picks?
Let's do this. So we've kind of glossed over, and actually maybe is a good theme for us, is to like dive right into the paper and then but return to some of the headwinds that we're seeing practice in science. So I know, Vincent, I was hoping to that we'd be together in vivo last week at the American Society for Virology meeting. I couldn't
Right.
make it, but I'm curious to get your sort of like leading up, I guess, long preamble to our picks of the week here. But what was your sense of virologists sort of being together as we're all kind of contending with the attacks on science that are currently underway in the U.S.?
Well, I have to say, so there are 1,400 people at the meeting, virologists, beautiful science being done, but, a lot of non US virologists there.
And I
Um,
should just say this meeting was in Montreal, right, up in
it was in
Canada.
Montreal. So, in fact,
Yeah.
I met a number of people who could never go to the US because
Mm,
of dumb restrictions, but they could
yeah.
go to Canada. And so,
Yeah.
Iranians, for example, can't come to the US, but they can go to Canada. And
Unbelievable.
so, there on the part of Americans, there's a lot of nervousness, right? Because they don't know what the future is going to hold for them. Um, but my takeaway was, Nels, I'm sitting in this audience with all these experts in a field, virology, none of whom are consulted by the current administration because they don't base their decisions on science. They base them
Yeah.
on their whims. And so we have multiple, not just virology, but there are multiple
Yeah.
fields where people study their butts off and figure things out and become experts in a field. And that expertise is no longer valued by this administration. And that's really the tragedy here: public health, epidemiology,
Yeah.
vaccinology, virology. All the expertise is being ignored. And what's going to be the outcome? Disaster. It's going to be
Yeah.
disastrous.
Yeah. Yeah. No, it's really interesting. So I was at a meeting maybe a week before ASV out near Boston, a smaller meeting, Gordon conference on the molecular mechanisms of evolution, and similar, yeah, grappling with a lot of this. I will say that one of the things I took away from it was, and especially from the trainees that were made earlier in their careers, grad students, postdocs, um, a real sense of community and energy and optimism somehow, even as as we're all navigating all of this garbage and worse. Um, and really found some inspiration in that as well. And so I guess that's kind of a long lead up to my science pick, which is a short essay that I just found a link to in Science Magazine. This is titled "My Academic Job Offer Was Rescinded. I'll keep going, but U.S. researchers are running out of road." And so this was a postdoc who went on the job market a couple of years and went as far as to have a job offer at one or two universities, and then in the decision-making process, learned that the offer was rescinded, just as they're sort of stepping through a lot of the regular, sort of chapters of life with kids and spouses, and how do you sort of balance all of these things. And so, anyway, it's a short essay. I think what really got me on it was both, um, sort of the headwinds, but also like a path forward here, and the idea of resilience and the power of like our individual stories. So, right, as we look at the whole system and the attacks on it, as it relates to virology, I think pretty recently we just saw pretty brutal decision making on what gain of function research looks like, with NIH grants pulled because of arbitrary and capricious interpretations of what gain of function research is and is not, and how that is just incredibly disruptive. The kind of things that could put like the work that we just talked about in complete jeopardy, depending on the situation and the timing for individuals, the individual scientists who are trying to practice this work. And so to really boil down to our own stories and to have the courage to share them, I thought was a really powerful sort of moment here. And so that's my science pick of the week. This is an essay. I think it's in the current issue of Science. Should be open access, and we'll put links in our show notes, et cetera. But the sort of plight of early career scientists.
So this is a person who had a job offer, a tenure track university job offer. It was rescinded. And so they said, "Oh, I can stay where I am." Nope, you can't. We don't have enough money. You can't stay here. And so what to do? And this individual goes through the thoughts, and in the end says, "I'm going to keep on working." And that's what resilience is, right? You can't give up. You have to keep on pushing. And I love that, that's the last sentence. That I've decided is what resilience really means.
Yep. I saw that at that meeting, sort of in a number of other individual stories. And it's really, really important. All kinds of crosswinds right now, and ways that you might get a grant, it gets pulled away, or you get one, it makes something else harder because of all of these entanglements that really aren't recognized by the people who are making these decisions currently. Okay, Vincent, what's your science pick of the week?
So I have a more mundane pick. It's science. It's an article in Nature it's a commentary in Nature by Xiaoji Lim. There's a new acid in our rain. Should we be worried? So there's a chemical called trifluoroacetic acid, which is used extensively in research and manufacturing. And the levels are going up all over the place. It's in rain, it's in the environment, it's in plants out there, and many other places. They have wonderful graphics on what is this chemical. It's kind of related to those forever chemicals that stick around and accumulate in different places. This one seems to be excreted very quickly from humans. So the levels are not high in humans, but they have some graphs about how it's increasing in the leaves and trees in Germany, groundwater samples in Denmark, ice cores in the Canadian Arctic. And the question is, if the more it increases in the environment, is it going to increase in us? There's no evidence that it causes any harm, but you don't know, right? That's the problem. Some people in this article say, it's just like salt, it's harmless, but I don't know. Hard to know.
Hard to know. These kind of uncontrolled experiments. I mean, the PFAS, the forever chemical, is an interesting link here. Actually, at this meeting I was at, there was a science hero of mine, a researcher at the University of Minnesota, Larry Wackett, who works on figuring out microbes or evolving microbes, directed evolution, or really doing the chemistry of, so in this case, developing microbes that can catalyze or degrade PFAS into harmless byproducts, right? And so yeah, again, as this is sort of one of the great reasons to support science is to try to come up with these
Yeah.
solutions to some of the problems, as opposed to sort of trying to ignore them, or worse, add to them without it, while at the same time taking away potential sort of technological interventions or in these cases. So yeah, just another one to add to the
So
radar.
the problem here is that these fluorine, chlorine, carbon bonds are very hard to break, and so this thing is not degraded, much like the PFAS is. So
Yeah.
basically, this looks like a small PFAS, which can persist, right? Because they're not degraded. Anyway, it's a good article. You should, if you can, you should check it out.
Yeah, cool.
All right, that'll do it for TWiEVO 116. A quick one here, but I think that's not bad. Let's see if we have any other notable. Hello, Noir. Noir has arrived,
Great.
and you can go back and see the recording. But thank you for your support of science
Oh, wow.
communication,
Yeah.
Noir. We really appreciate that. I want to
Thank
thank
you.
our moderators today for helping us out. We had Vanity Nutrition and Barb Mack. And for all of you coming and listening, if you want more live stream, 8 p.m. tonight at this same place, I'll have office hours with my special guest, Angela Rasmussen. You can find TWiEVO at microbe.tv/TWiEVO. And again, consider supporting us, microbe.tv/contribute. Nels Eldy is at cellvolution.org and L Earlybird on Blue Sky. Thank you, Nels.
Hey, thank you, Vincent. And we didn't meet up at this meeting, but we'll catch up in vivo at the next one. We'll be back in about a month for TWiEVO 117. We'll see you all then.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, a podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, stay curious. Bye-bye.